We can move to our next speaker, Professor Heinrich Haas. He is the Chief Technology Officer at Neovac and has a vast background both in academia and pharmaceutical industry. He has developed nanoparticle products to clinical stage and focuses on advanced approaches for nanoparticle development and control. He will discuss the relevance of supramolecular organization for quality and activity of mRNA LNPs.
Hi. Yeah, thank you very much for this kind introduction. Thank you for the organizers to allow me to contribute a little bit further insights into LNP and pharmaceutical development. So with the introduction of Dan, I can add another aspect which is more on the molecular and on the physical side of these formulations. And as said, I'm basically trying to span the scope from real basic research to practical pharmaceutical development, which is sometimes not so easy because you have to bring together quite different personalities. But in the end, it is worthwhile to do it because this really brings innovation into the field. So I will report a little bit about Neovac as a company, as introduced also by Dan, and then I'll show some more basic research results done together with the university in Mainz with the Department of Pharmaceutical Technology. So maybe one of these guys you have seen just before. This is about Neovac, co-founded by Dan and Professor Sir Adrian Hill. In the field of LNPs and vaccines for novel mRNA-based therapeutics. Very briefly, it's just one thing. We have seen lots of lipids and LNPs. This is a slide we made to represent that from a more general perspective. On the left-hand side, you see the classical known ionizable lipids like the aniline one and the acritas one. And they all are characterized by a certain architecture, namely being built up of a hydrophilic head group and the lipophilic tail, more or less stimulated by phospholipids. And the value of the LNP and lipid technology developed in Dan's lab is that we have introduced more structural and functional elements. So we can expand the lipid architecture or the molecular architecture by structure elements, by functional elements, and by responsive elements. And so this is more or less being also stimulated by other delivery systems, dendrimers, polymers, and so on. And we then can tailor and engineer our lipids and our nanoparticles to achieve certain biological activity in the sense of modulation of biodistribution, organ targeting. We have heard about that. We can modulate pharmacokinetics by introducing certain cleavable elements, for example, by tailoring biodegradability, and we also can tailor stability. Just one thing which we would like to share with you is results from a recently successfully finalized clinical study using COVID as a model antigen, which was done in UK together with Jenner Institute. Intention was to investigate safety and tolerability of 3 doses, 25, 50, and 100 micrograms, just as in the light of what is known for the COVID vaccines. And here the take-home message is that as others, we can demonstrate efficacy for vaccination, but the impressive difference is that we could demonstrate quite an improved safety profile of this LNPs. And this is basically the discriminating factor. Here you see a cartoon with comparison of the Neovac LNPs with the two licensed one from BioNTech and Moderna, and this is just the side effects. In a nutshell, both the frequency and the severity of the side effects is much lower than the two licensed products. And we are convinced that for future products, this will be the discriminating factor because basically with immune titers, we are all on the safe side. But the safety is that what makes development of a future product successful or not, because this is so much in the media, justified and non-justified, but some concerns are also justified. And so we are really convinced that with such a profile, we have founded the baseline for coming up with novel products in the field of vaccination. This can be COVID, but now we are focusing on other indications. We will share with you more details once this has become into reality, but it will be basically applicable to all viral infections in general, optionally also to bacterial infections and other indications as mentioned by Dan. So that's just, let's say, as an eye catcher for you to have a look at us in the future when we'll try to publish that and bring more news about that into the community. Now I would like to switch gear a little bit and have a focus a bit more on the topics of Bia Sartorius and the separation and analytical techniques. This is done in collaboration with the Department of Pharmaceutical Development, Technology at the Mainz University, headed by Peter Langgut. And you see us here somewhere, that's the globe with this I show him if I'm traveling in China. And we are basically in the middle of Germany in the Rhine-Main area with lots of relevant universities, research institutions, and companies all around. So, and the way I look at this here, I share with you here, is a more general perspective. What are we doing if we are making nanoparticles? We bring together the messenger RNA with any kind of excipient, which can be lipids or lipid mixtures or polymers, and then by a certain process of self-assembly, nanoparticles are formed, which can be quite different in their architecture and in their properties. Here you see three examples. The top one is the classical lipoplex approach, which results in these lamellar structures. We'll show you some data on that. This is the foundation for the BioNTech cancer immunotherapies. This is the LNPs, which you all know, where you mix an ethanol solution of lipids together with RNA. And you can also mix a polymer in water with RNAs, which is also a polymer, and come to nanoparticles. All of them are self-assembly protocols, and I would like to stress a little bit the foundation of these self-assembly protocols, which can be understood, and this understanding can be used to do the nanoparticle engineering in a controlled manner, and it's also important for the quality control of the particles which you have formed. So it's more or less an introduction of concepts which can be helpful for you in your daily life. And in principle, everything which I will present from here on is publicly available, and you are invited and encouraged to use that at home. So here you see a typical panel of quality control assays for lipid nanoparticles. On the left side, the ones for RNA, where you have your identity, integrity, residual double-stranded RNA, and so on. So this is basically derived from the biopharmaceutical biomanufacturing space. On the right side, the typical assays for the drug product, the LNPs, and basically you see the repetition of the RNA controls and a little bit of particle size, maybe free and bound RNA. But the problem with that panel of QC assays is that it completely neglects the colloidal nature of the nanoparticles. So nanoparticles are colloids. They are consisting of a large number of different molecules which self-assemble driven by electrostatic and other forces. They can assume different types of structure. They can exist in different phases, as shown here with the lipids. So there is lipid polymorphism, and they can also be observed in the terms of polymers. So this is here you see the typical way how biochemists draw the messenger RNA just as a block of different functional and structural elements. but in a different way, you can see that as a polyelectrolyte. So RNA is a very large, very highly charged biological macromolecule, and this drives certain properties. And in the end, to the right side, you see the typical cartoon of the LNP, and also for the LNP, further to dynamic light scattering, which gives you something similar to particle size. The quality control does not cover so much the certain properties which could be relevant for biological activity, and we will follow up with that in the next slides. So one thing which nicely demonstrates how you can use insights from colloidal sciences for particle engineering is how we made ultra-small self-amplifying RNA nanoparticles with a polymer. And this is driven by research which has been done years ago in the interfacial and colloidal sciences, where people tried to investigate self-assembly between oppositely charged polyelectrolytes. You can do a layer-by-layer adsorption of, in that case, this was also involving PEI, which is used for transfection and anionic polymers, and you can study here all kinds of parameters of the systems. And what has also been shown is that you can use PEI, the transfection reagent, with polystyrene sulfonate and study particle formation. And the physics, the underlying physics of the systems is well described and fully understood. And this you can take as a basis to advance a little bit the technologies for making polyplex nanoparticles with PEI. PEI is quite well established for DNA transfection. And it has reached clinical stage for DNA delivery systems. It's widely used in vitro as transfection reagents. And I will show you how you can control the self-assembly properties of these systems, and by that decide which type of particles you make. So you can make usual polyplex nanoparticles, which are large in the tens or hundreds of nanometer scale, but you can also make small particles which basically consist only of a single individual or siRNA copy, which is complex by PEI. And this has been demonstrated, for example, by analytical assays. This is kind of orthogonal to the chromatography, which is done by Beer. You see here two techniques: asymmetrical flow field flow fractionation, which separates the particles as a function of their diffusion coefficient. And this is analytical ultracentrifugation, which separates the particles by a combination of diffusion and density of the particles. What you see is that if you do the separation technique, so have a channel where the particles flow and the cross flow, and you see that if you investigate this polyplexes, you see different fractions of rather monodisperse entities, particles. Where if you increase the N2P ratio, this is the excess of the polymer, you shift this distribution to the first single peak which shows up. And here you see the pure naked RNA, which is not complex, and this is much broader. This reflects the organization of the RNA as a random coil with a relatively broad size distribution. So what we do is, by complexing, we transform this broad shape of illusion profile to single peaks, and the same you can see with analytical ultracentrifugation. Another method which I would like to share with you, which I consider important for investigating such systems, is scattering techniques, X-ray scattering in that case. And if you do that, you measure curves which look like that, basically relatively boring, but you can analyze them quantitatively, and you can calculate a parameter which is called the pair distribution function. And this shows you how big the particles are and what their internal organization is. And if we do this complexation assay with the polymers which we applied, we can transform the RNA into a very, very compact status, which is having, in this siRNA case, 3 million Dalton, a size of less than 30 nanometers. So this is basically close to the physical lower limit of compaction of the particles. This is because the polymer has such a low molecular weight per charge that it doesn't contribute so much to it. And the conventional RNA has a completely different organization. This is if you add lots of sodium chloride with the RNAs, you screen the electrostatic repulsion, you make it more compact, but by far not as it's possible by the polymer. And this is the RNA in water. It's really extended random coil organization. So this another technique which is useful is Newton scattering, which gives you additional information on the composition and the shape of these particles. Simply spoken, you can highlight different molecular moieties in the particles. What you do is you do this Newton scattering experiments with different D2O to H2O mixtures, and you get scattering curves, and you can do an analysis which is called Guinea analysis. I will come back to that. And by a so-called contrast matching analysis, you can find out how much of polymer and how much of RNAs in your particles. The further analysis is what people call a Sturman plot, and by that you can analyze if this particle has a core shell organization or if it's kind of anisotropic, like an ellipsoid and so on. And this gives you supplementary information for complex particles. And in a nutshell, why is this interesting? It's interesting because the single particles, they have a tremendously increased biological activity compared to the classical polyplex nanoparticles. What you see here is an experiment with using Lucifer's expression as a readout, as a model RNA. And we use always the same amount of RNA and added the excess of polymer. And keeping the RNA constant, you see that the luminescence increase as a function of the PEI, the polymer excess. And basically, it's a linear increase, and we do think that this is due to the transformation of large particles into individually single RNA molecules which are complexed by the polymer. And this could be also reproduced by, for example, a vaccine experiment. Here you see another example where we completely where we have the same dose and reduce the dose. And what comes out is that by reducing the dose, we can basically maintain the activity constant. In other words, this tells you that the classical experiments were done in saturation. And if you go to the analytical route, you can go to doses as low as 50 nanograms and get pretty nice vaccine titers both for antibody and cellular response. And this allows you to reduce the dose, reduce the toxicity, and increase the activity. And this is just by applying certain concepts of physical chemistry, of colloidal sciences into particle engineering for making polyplex nanoparticles. That just recently has been published in Nature Nanotechnology after long fights with one of our referees. And here you see how the polymer condenses the RNA to this very small magic bullets, as many years ago described by Paul Ehrlich. And now, expanding that a little bit, I would like to dig a little bit deeper into control strategies for mRNA nanoparticle systems. So I showed you that we do think that scattering techniques, X-ray scattering, can be very useful to learn something on the structure. Here you see scattering experiments for polyplex nanoparticles and LNPs. You see scattering curves, and the first thing by the eye, you see that they are different. And one application can be that you just look at the curves and compare them. For example, if you make an upscaling or shift, go to another manufacturing site and want to compare the quality of products manufactured at different sites. But this becomes much more powerful if you have measures in hand which allow you quantitative and comparable analysis of these curves. And we have just recently published a paper where we proposed a certain strategy of data analysis regarding 3 different ranges of this small angle scattering curves, where we analyze the Guinea range, we do a Poirot analysis, and we analyze the peak. Just to introduce that to you, if you plot the low Q region of your scattering curves in a logarithmic manner as a function of Q square, you get kind of a Guinea plot. And this shows you basically a linear correlation where the slope gives you directly the radius of gyration of your particles, irrespective of shape and anything else. And you also can analyze the extrapolated value to the zero-angle scattering. This gives you a value which is correlated to the number of particles, to the volume, and to the contrast of them. And this can be extremely helpful to quantitatively analyze curves. I will show you that. So another parameter which is not so much regarded in the community is the fractal dimensions of your particles. We have seen indication that particles with a higher fractal dimension have a higher transfection efficacy in vivo, and we think that this is due to elevated, improved interaction with the endosomal membrane, and this is favorable for endosomal release. Although this you find more described in more detail in our paper. And finally, if you look at the peak, you can analyze the peak position, which gives you the repeat distance of your structures. You can analyze the peak width, which gives you the number of layers. And you can analyze the area of this peak, and this gives you the amount of material which is present in that type of order. This is something everybody can do with scattering curves generated in different laboratories, and then you can compare quality from different groups and companies. And now coming back to particle size and flow field flow fractionation, the particle size of lipid nanoparticles is basically still neglected in quality control approaches because you just use a method which is quite well defined, but it gives you only a very limited insight into the real physical properties and the real physical size distribution of the particles. And we are quite keen on using this AF4 technique. So it separates, as said, particles according to a diffusion coefficient. And if doing so, you can, if you have here an LNP or lipoplex formulation, you can separate free RNA from the particles. You can use multi-angle light scattering or dynamic light scattering to measure the size of the particles, which nicely increases as a function of the elution time. You can analyze different parameters like the radius of gyration versus radius of hydration and get information on shape characteristics. So that's already a quite good improvement, but what is still missing is the quantitative insight. So here you see like an elution protocol, and so nicely particles size increases as a function of elution time, but what you do not know is how many particles do I have at a certain size fraction. And we solved that by hooking up the AF4 to a synchrotron source, where we did further to the classical light scattering and UV measurements, small angle scattering, and combined the signals from all these measurements. So this is the readout, which if you look at small angle scattering of X-rays in combination with light scattering and UV spectroscopy, again, you can separate the free RNA from the particles. But what you can do in addition, you can measure the scattering curves of the free RNA. We saw that before, that you can analyze, get information on the conformation of the RNA, and you get information on the size separated particles. And here you see this Bragg peak evolution as a function of elution time. And this helps you to get quantitative insight into the particles. And to make this more concrete and clear, here you see measuring an experiment with measuring LNPs. Again, you see the typical peak of the particles. And what people very often observe is a kind of shouldered, longer, higher elution times, and nobody knows if this is good or bad. Is this now a degraded product, or can I neglect it, or do I have to throw away my batch? And our data helps you to get an information on the relevance of that observation for the quality. Again, here you see that the usual analysis of light scattering in combination with X-ray scattering. These are the scattering curves over the elution time, and you see you always see particle-like structures. At a certain stage, the peaks come up, and then they vanish again. So what you now do, and this is fundamental for this method, for each individual of these scattering curves as a function at a certain elution time, you do that Gini analysis. From the Gini analysis, you get directly the size of the particles or the radius of gyration. From the radius of gyration, I can calculate a volume, assuming a certain shape. And then I can combine that with my extrapolated zero-angle scattering intensity. So I have then this value, I have the intensity, I have the volume. I do know the contrast because this is known from the molecular properties, and from that I can calculate my particle number. And having this information, I can calculate the absolute concentration, right, thank you, the absolute concentration of the particles at each elution time and at each size fraction. And in doing that and several other analyses, I have now, from my measurement, originally an allogram where I get all kinds of parameters as a function of time. I get now my absolute size, and from that, this I can correlate with the particle number, structure, and everything. And so I can calculate absolute size distribution profiles. I can get all kind of structure parameters as a function of size. And this helps me to analyze the particles quantitatively, and I can come to a conclusion if this additional feature which we saw in the allogram is relevant or not. In that case, actually, it was not because it was just overestimated due to the high scattering activity of these particles. So in a nutshell, as I have to finish, so this is just an approach to introduce more physical, chemical, and biophysical methods into, first of all, concepts for designing and analyzing nanoparticles, and we do think that this is very valuable to bring future products into reality. Just to add up, there's a very nice conference series where we talk quite a bit about these biophysical methods. Next one next year in June in Strasbourg in France, and so if you're interested, you are invited to join that and share your results and get also impact from others. That I would thank to some of our collaborators, and I'm happy to take questions if we have time.
Thank you very much, Professor Haas. So we're open for questions.
It's not an invited question, but so in Sachs, in wax, you showed very nicely the length, the distance between the layers and the nucleic acid, the lipid and the nucleic acid, which you measure. What happens with LNPs? So this is predominantly seems to be very nice for lipoplexes.
This is LNPs.
This is LNPs, okay. So you can see the distance in layers because it's like inverted micelles. And then there are...
No, it's not. So that's a long discussion in the community. We do think it's not inverted micelles because if you have micelles, you don't have a repeat distance. But right. We do have a repeat distance. So this means you need at least 2 or 3 of the same aggregates organized with each other.
So the helper lipid here plays a very important role, no?
Yeah,
because
I mean...
if the percentage of the helper lipid exceed a threshold, it will be forming repeated kind of bilayer structure. Or do you think that the helper lipid is less important?
Yeah, the helper lipid is of course important, and some of the helper lipids may like phase separate, and then you see 2 different structures. And we have indication that this is the case for certain of nanoparticles also that is published. So what you see always is just the structure. So you don't know what the structure comes from. You see a repeat distance, and you see how many blocks are organized with each other, with respect to each other. And then you come up with your interpretation. This is repeating lipid and RNA multi-layers or whatever you think it is. And of course, what you also can reveal is if you do this more carefully, you can get quite nice information on the core shell organization. People have done that. So like enrichment of cholesterol or DSPC at the outer shell and so on.
I've been looking for an image which you then showed in one of the slides, which was your Sachs measurement of mRNA in presence of different salts. So thank you for bringing it here. And I think it's very exciting. It bridges the mRNA world and the LNP world, which need to talk more to each other. And you show a dramatic change in gyration radius between no salt and even a little bit of salt. I'd be very interested to know what half molar sodium chloride does. Presumably it compacts RNA even more, because that it means that when we perform oligo-DT purification of mRNA, our RNA is literally doing a harmonica type movement. It's dancing a lot, and I think this plot helps us a lot in purification of mRNA.
That's why I think that's a good data set to show here, because it shows you how different the organization is as a function of the ionic strength. So first of all, you have to discriminate monovalent ions by divalent ions, so I have a big difference. In that case, the reason why this particle here is so small is that the polymer, the PI, is a polyelectrolyte, so you have many charges all together. While here you just have single monovalent charges, so the energy threshold for a counter ion to exchange with the bulk phase is very low. But here you have a very high energy barrier for if you want to get the PEI out, you have to break like 500 electrostatic charges at the same time just by thermal fluctuations, which is virtually impossible. So you make these particles, and once they are formed, they are also stable in circulation. That's also good for the biological side. Only if you bring it in contact with, let's say, an oppositely charged membrane, so then you can get it apart. So, this is, I think it's really, really instructive on how different the conformation of your RNA can be as a function of your process conditions. And you can take this into consideration for planning your processes. That's why these data are so useful. And you just do it once, and then you have understood it, and you know what you need to do. You don't need to do this, let's say, every time I make a new manufacturing batch.
But this depends on the size of the RNA molecule and the sequence as well. Because small size probably will behave differently than a very large one.
Yes, but the differences are only if you go to very, very small sizes. So I don't think that the physics between messenger RNA and self-amplifying RNA is different. There is a difference between silencing RNA, siRNA, because this is just 20 mirrors. And then again, the enthalpic and the entropic forces are completely different. But here, the bottom line is you have to consider interactions between oppositely charged polyelectrolytes for understanding that. And polyelectrolytes interact differently with a polymer than monovalent and divalent ions. And this goes basically beyond the basic secondary structure. This also contributes, but these are more the cross effects. So this will happen always irrespective of detailed differences in secondary structure.
Okay, thank you, Professor Haas. So now you're invited to lunch. Please follow up our colleagues who will show you to the cafeteria, and we will resume at 1:45.